Thank you.
Okay, thanks, Beth. That's going to be really hard to follow. So appreciate all those really big picture ideas, and I'm going to literally take you guys down in the muck into the sedimentary habitats and talk about who lives there. So one of our goals is estimating the distribution of sedimentary benthic habitats on the eastern Pacific shelf broadly, and then I'm going to take you to our project site and see if we can detect some project effects. And this is important to say that we're looking at the distribution of sedimentary habitats and not just sediment, because I could tell you that in one slide. Sediment's almost everywhere in the Pacific Northwest. Rock is hard to find. But what's more than just sand? So to do this, we conducted a regional survey where we did high-resolution mapping, sediment ground truthing, did a bunch of animal collections in sedimentary habitats. We also had an ROV component to this study at the few places there were rocks, and our approach was very similar to Antonio's talk, so thanks, Antonio, for that. And like I was saying, the objective is to map habitat, not just geology. And eventually we use this to develop predictive capabilities of where to find high-priority habitat or species, or predict changes to species if we see changes in the habitat distribution. So the high-resolution mapping was conducted by my collaborator, Chris Goldfinger, so if you want to know details about how many kilohertz, I don't know him, but I can get you in touch with the guy who does. Ground truth with grab samples, so look at the acoustic backscatter, find high transition places to further ground truth what that signal is telling you about the sediment. And then finally, make habitat maps. And so the first step of this is just based on lithology. And so what the mapping lab does is they take the data from our surveys and then go into the various different kinds of databases that are available and get more sediment data. And make a map that we've got mean grain size here, and then we've got percent sand. And you can see that the sand is in 10% bins. So showing that 10% sand, and 20% sand, and 30% sand. But we don't know if that is at all meaningful to the organisms. And so after the mapping was completed, my team went out and we took 153 grab samples between Northern California and Washington. And we take CTD casts to get water quality. We take subsamples of the sediment and do grain size, percent fines, total organic carbon and total nitrogen. Then we get the organisms out and we identified 139 different species from the grab samples. And then we do a lot of different kinds of multivariate analysis, the final of which is this LINKTREE, which is a routine and primer, and I can walk anyone through it offline if you want to ask questions. But it's an iterative splitting of the stations based on the organisms that live there and the correlating environmental variables. So basically, the first thing that splits off as really unique are sites that have a lot of gravel in them. And then what splits off are sites that have more than 10% but less than 60% gravel. And then it gets really interesting. So the next split off was less than 84% sand, and many stations all grouped together in less than 84% sand. So that was a fairly homogeneous environment. And then when you get into high sand environments, you get a lot of branching based on depth, so a split in the 80s. I didn't update that, that's supposed to be in the 90s. And then further splits with grain size and depth as you get down the line. So in summary, what we found in this region is really two gravel habitats, a muddy sand habitat that had a fairly homogeneous species assemblage throughout it, deep sand, which was what we're calling rather homogeneous, so it was sand that was greater than 86 meters, we only saw one further depth split, and then shallow sand had enormously high spatial heterogeneity, with stations splitting out based on their organism distributions within really fine breaks in grain size and depth. And then finally, at the bottom of the tree, we find that places that have less than 99.2% sand are different than places that have 100% sand. So a very, very small percentage of fines actually resulted in significantly different species assemblages. This was a story we saw over and over and over again. So, just for part 2 of the talk, keep in mind that the places I'm going to talk about for part 2 are shallow, high grain size, high percent sand, and then shallow, smaller grain size, nearly 100% sand stations. So when we're talking about the distribution of sedimentary habitats on the shelf, basically what we found are species assemblages within the study zone are shaped primarily by percent sand, with secondary differentiation based on grain size and depth. And so instead of this 10% bin sand map that is just purely geology, basically what we need to do are adjust these bins to be reflective of species preferences, and then you actually have a habitat map. So that might look something like this, where really you've only got 3 or 4 colors instead of 10 colors of sand. Okay, so now we're going to zoom in to one particular place, and that's NIMRIC's, the Northwest National Marine Renewable Energy Center's Pacific Marine Energy Test Site, the North Energy Test Site. And so from now on, I'm going to call that NETS, the North Energy Test Site. Save all those other letters. And so what we have at NETS is a pre-permitted place for developers to come test scaled wave energy devices. And the way they test a scaled wave energy device is, rather than plug into the grid, they plug into our analysis platform, which we call the Ocean Sentinel. And so it can provide all the features of the grid at much lower power levels. So I've been doing baseline surveys at this site since 2010, and we've taken a lot of box cores, like in the regional survey. We've also done trawling to sample fish, which I'm not going to get to talk about in the 15-minute slot. And we've done a number of video surveys along the way. And so it's this grid of stations that's around the permitted test square. And in terms of the sediment and Infauna, what we've found is that the sediment conditions at the site have been extremely stable. And you can also see that we have 2 kind of different habitats. We've got a smaller grain size habitat at 30 meters depth, and then larger grain size habitat, which is with slightly more variability at 40 and 50 meters depth. And those 40 and 50 meters are pretty similar, statistically indistinguishable. And so all this time, this grid of stations that I've been sampling, we haven't seen any changes in the sediment, and we've seen very stable infaunal populations as well. And they've been stable over time, but they have very high spatial heterogeneity, as you might expect from our regional survey, because this is an area of different grain sizes, sand that's relatively shallow. And so the 30-meter stations are all similar, but the 40-meter stations, some of them look more like 30, some 40s and 50s are the same, and some 50-meter stations are unique. And it's been amazing that those distinctions have held true, I think, over 4 years. So, what happens when we put the wave energy device? We had Wet-NZ test in summer of 2012, and we had the Ocean Sentinel on. So we had a total of 8 anchors and 2 devices. And we didn't see any changes at this grid of reference stations, the closest of which was within a half a kilometer away from the installation. But when we took an ROV down and took it around the anchors of the devices, we did see scour, as you might expect. I don't know if you guys can see, but that's a sort of a shadow under there. So sediment's been scoured away from under the anchor. And this accumulation of shell hash in pockets all around the anchors, very near field. And so no one really likes me to drop a box core around mooring lines and electrical cables. That idea does not go over well. But in 2013, after they removed the Ocean Sentinel, they just unhooked it from the anchors and left the anchors in there. So now I had these things that I could approach with the boat, drop heavy equipment on, and no one cared. So that's great. So what we did is we got a range finder, and we used the marker buoy as an estimate of where the anchor would be. And basically dropped a box core, took a sample, backed off, dropped the box core, took a sample, and did transects away from the anchor to see if we could quantify any of these changes we thought we'd seen on the ROV. Okay, so if you thought that Gail was a little bit tentative in talking about device interactions yesterday, this is some like really large hand waving because I've only done it twice. So I have an N of 2, and we're all scientists, and everyone said never measure anything twice, right? You got to have at least 3. But this is a new project, so we've been out twice. Okay, so when I pull the data from October 2013 and April 2014, so here we've got, this is the average of our 40-meter stations that we've always gone and visited. This is from the Ocean Sentinel's northeast anchor, doing a transect heading east, so heading inshore, where we would expect to see effects of the anchor. This is from the westernmost anchor headed west, so offshore, where we wouldn't necessarily expect to see effects. And here's the 50-meter station that's about a half a kilometer away from those anchors. And so, you know, we've got one really shell hashy sample, but the mean here from the eastern anchor is higher than the means from the reference stations. So it looks like I was able to quantify that accumulation of shell hash around the anchor. Okay, so do we see effects on grain size? Well, maybe. I have an almost significant p-value here. So here's the same thing. And you remember 40 and 50 meters are generally not different. And it looks like moving inshore of the anchor, we're seeing some more scour realized in an increase in median grain size around the anchor. Head waving. And so the question is, are we seeing effect on the organisms? Remember, this is a kind of site where differences in depth and median grain size and percent fines can have a really large effect on changing the assemblages. Well, since we took these data last week, I haven't actually had my staff identify every single organism that we've gotten. But what it looks like is when I went out in the fall, we definitely got fewer organisms at the anchor station than we got at the 2 reference stations nearby, 40 meters. But then when we went out last week, not so much. It's looking pretty much like the reference stations. And so I have a hypothesis I'll pitch to you guys, and you can choose to accept it or not. So at the end of the summer, things have been pretty stable, and so the effect of the anchor may be relatively more important. But now that we finally got out in April, after attempting to go out in December and January and February and not making it because we had really big storms, and things are getting all mixed up, that maybe the signal of the anchor has gone away because everything's getting all mixed up. So that's my so, in summary, species assemblages offshore within the region are primarily shaped by percent sand and depth, with finer differentiation based on grain size. The North Energy test site mostly consists of 99 to 100% medium to coarse sand, so there's even little capacity for change due to scour. There's no fines there in the first place. How much change can you expect to see in terms of the loss of fines? We're really only looking at a grain size change. So there was some evidence of shell hash accumulation and scour on the anchors, with potential reductions in infaunal abundances at the end of the summer, but little evidence for anchor effects at the end of winter. And so stay tuned. We just got this week our permits to leave the anchors in through the end of August. So we'll keep going out and see over the seasons if we can track any changes. And we didn't detect any effects greater than a half a kilometer away from this installation. So anything that I may or may not observe right next to the anchor is certainly not being appreciated in our field. And with that, I'd like to acknowledge all of the support it takes to collect and process almost 500 box scores, travel in terms of money and logistics from these people, and my husband Will, who's at home by himself with a toddler for a week, so that I can join you guys. So.
Again, if we've got one burning question, prepared to take it. Anybody? Okay, we'll leave it there. Thank you, Sarah. And I'll ask Anna Redden to come up, please.